Hello and welcome back to another episode. So in response to some of my students' needs and special requests, I'm going to do a photosynthesis and cellular respiration recap summary lesson. This one should probably not run for as long as the others because I'm really only just going to touch on the important information, what is really important for Year 11 biology, and the things that are that will carry over into Year 12, but specifics looking at the study design rather than just going into all details. It'd be a good idea to watch this video first and then look at the other videos possibly. So I might try and organize that somehow in my playlist. I'll see if I can figure out YouTube's way of doing that. But hey, this is how we're going to do it. And we're going to crack on from here. So having a look at the equation for photosynthesis real quickly, if we're accounting, the black dots, carbon, oxygen, hydrogen, there's your colors, we have our 6 carbon dioxide molecules and our 12 water molecules coming into the equation. Now we know also that photosynthesis, light energy, we're going to get those light photons which are causing the reaction to happen, like the catalyst, the energy, exciting the electrons in the electron transport chain part of things. But I'm not going to go into those details, okay? You can watch those in the other lessons. But what we'll talk about is where all these molecules are accounted for. So when the reaction happens, we have a shift. And there's our 6 oxygen molecules are accounted for through those through the oxygen that they were left over there, our 6 water molecules that are left over as part of the reaction as well, and the main event, the main molecule that was being created by photosynthesis, which is our glucose C6H12O6 molecule. And this is really important. You do need to know what the equation for photosynthesis is. You do need to know that it's 12 waters here and 6 waters over here. You do need to know that the reaction is triggered by light energy. There's lots of information that is important to know. And in terms of this equation, actually, no, I'll take that back. There's not lots of information that's important to know. Really, it's just the equation in all of its splendor. All right. So you guys have probably seen this model before. It was definitely in the photosynthesis lesson, the longer version of it. I've used it for my students in class, and we're going to be talking about this model in its entirety, but just to make sure that we're aware of what we are looking at here. We are looking at the chloroplast, sorry, I'll write it up here. Chloroplast. Perfect. This is our outer membrane. Our inner membrane. That line there. But most importantly is this is our granum. So the stack. Grana, and an individual disc where everything is taking place is going to be our thylakoid. This reaction over here is happening in the stroma. Okay. That's it. That's the important labels for the chloroplast. We don't need to go into all of the other bits and pieces of detail at this stage, as long as you know that this reaction over here, the one where the light is coming in, is happening in the thylakoid, where there's all the chlorophyll. As long as you know that this reaction over here is happening in the stroma, the soupy liquid of the chloroplast, then you are off to a flying start. All right, focusing in on the light-dependent reaction, we know that at this stage, if we go through this, there's the electron transport chain happening across the membrane of the lumen here. But really, what we just need to realize is that at this stage, this is where our water is coming in. Okay, our water molecules are coming into the thylakoid here. Our light photons are coming into the thylakoid, as well, providing the energy to excite our hydrogens, electrons into going through the electron transport chain. The major things to remember about this is what is being created that carries over to the next reaction. We do have ATP that is being created, and we do have NADH These two molecules here are our, well, they're classified as coenzymes. They're our carrier molecules, yeah? That's their role. Their job is to carry, ATP's job is to carry energy, while these guys' job is to carry electrons and to an extent our hydrogen ions as well. These guys here, that's their job. They're going to carry these over to the next reaction. And this is the important part of the light-dependent reaction. This is what we're getting out of it, the ATP and the NADPH. The byproduct, of course, being created down here is our oxygen molecule. And it's a bit big, but yeah, it's O2 molecule. And it'd be a bit silly if I tried to write it in red inside there. But hey, this is what we're meant to get out of the light-dependent reaction. If you can go ahead and explain that there's an electron transport chain going on, if you can explain that there's, the hydrogen ion gradient between the two membranes, which then allows the hydrogens to re-enter the lumen, to then be picked up and form water molecules as a result here as well. But also to recognize that the ATP synthase protein is what's spinning around there to get our ATP via that, the hydrogen moving through it. Great. If you can explain those things, fantastic. But in all seriousness, this here for the light-dependent reaction, this is the important. Knowing that in the thylakoid, water molecules are going to come in, light molecules are going to come in. They're going to be split. We're going to get some ATP and we're going to get some NADPH as carrier molecules to go to the next reaction, and that oxygen is your waste product. That's it. That is the most important stuff to get out of this. So then it moves very quickly into the light-independent reaction. What happens with that information? What happens with that stuff? So remember that we had ATP coming in, and we also have NADPH coming in. They come into this reaction here, which also has CO2 entering it at the top. The only other thing that I could possibly mention is, the importance of the carrier molecule, or the molecule Ribisco, because it is classified, I believe, as a coenzyme. There might be some questions about it, but really that is the only thing that you might want to add up here, is that Ribisco is the first molecule that gets grabbed hold of, or was created from the CO2 entering the Calvin cycle. Oof, I forgot the word there. Better write it down. So this one here, Calvin cycle. And then knowing the 3 stages of the Calvin cycle, knowing that Ribisco is the molecule there, yeah, great. But also knowing the 3 stages of the Calvin cycle, knowing that the first stage here is is your carbon fixation stage. Knowing that you're also going to end up getting a reduction stage. And then knowing that you also have a regeneration stage. Those 3 words in the cycle is all that we really need to know. Knowing that we have, X amount of the ATP that comes in, knowing that we have X amount of the NADPH coming in, I don't believe that's something that's, always going to pop up. But if you go back and have a look at the video or if you end up looking at the video for the photosynthesis reaction, you'll be able to see how many NADPH and how many ATP actually come into this reaction to be able to produce our molecule. So we end up with a G3P molecule, which eventually goes and forms our glucose, our sugars. So we get glucose out of the Calvin cycle here. Now we also have our unloaded carriers up the top. NADP plus and ADP plus, and this guy here is PI, so that's just a little phosphate on his own. So these guys here are unloaded carrier molecules. They don't currently have the electrons, they don't have the energy in them at the moment, and so they're getting sent back to the last reaction, back to the light dependent stage to become loaded up again, so they can continue this cycle round and round and round and continue to bring energy into this part of the cycle, the Calvin cycle. That's it. That's the important information for photosynthesis. Yeah, I went into a lot of detail in the last video, and there was a lot of information. But if you guys have this filled out, you know, there's your sugar, your CH2O. But you know, when you put two of them together, and there's lots of them going around, great. But yes, you have your cycles there, and that's the most important thing. Which brings us to the next one. Cellular respiration. We've seen this happen before, the reverse of photosynthesis, where photosynthesis produced the 6 oxygen and the glucose. Cellular respiration uses it. Cellular respiration uses the products of photosynthesis, even the byproduct of photosynthesis, exactly, in its reaction to create 6 carbon dioxide molecules as a waste, 6 water molecules as a waste, but the main event is getting the 36 to 38 ATP out of it. So each stage needs to be described. You need to be aware of the stages of cellular respiration, but you don't need to know the intricacies, the nitty-gritty bits and pieces. Again, I went into a lot of detail in my last lesson that I did for this, so it was a really, really long video. But cellular respiration can be described in 3 stages. Stage 1 is glycolysis, so the cutting of, glyco, meaning sugars, and lysis, meaning to cut. So we're cutting sugar in this first stage, and we start off with that glucose molecule. It goes through, 8 to 10 chemical reactions to be cut up into 2 pyruvate molecules. Essentially, this is a 6 carbon molecule. These guys are just 3 carbon molecules. Yeah, they just cut them in half. So this is glycolysis. That's as simple as it is. What we also get out of this reaction is we get a net of 2 ATP as well. Now this is a net of 2 ATP. I describe it in the video as well why that's a net of 2 ATP. You actually get 4 ATP, but because 2 ATP are used in the reactions, it's a net gain of 2 ATP. As well as getting 2 ATP, you also get 2 and, oh, come on pen, there we go, ADHs. NADH is the similar carrier molecule, but in the as NADPH, it's just that it exists inside the mitochondria instead of the chloroplast. This guy here, again, it's an electron carrier. That's its role. It's going to shuttle or carry electrons to another stage in cellular respiration. All right. So here is where we have to check for oxygen. Check for oxygen. Oxygen is present. Great. It's time to move from the cytosol, where this is happening. So this is happening outside of the mitochondria in the cytosol or cytoplasm, into the mitochondria itself for stage number 2, which is the KREB'S cycle or citric acid cycle. again, describe it in the longer video, but the Krebs cycle happens in the mitochondrial matrix. Again, the soupy fluid solution inside the mitochondria. And yeah, it's called the citric acid cycle because one of the products, one of the carriers of all the carbon inside this cycle here is citric acid. So in the process of coming into the cell, the pyruvate changes to enter the Krebs cycle. It changes into acetyl coenzyme A, or acetyl-CoA. Now we get two of these because we get two pyruvate molecules, so there is two of these acetyl-CoAs, and they enter the Krebs cycle. And they go ahead and produce a mass amount of electron carriers. In the process of getting them into the cell and turning them into acetyl-CoA, I figured out the other day, and this one's pretty good, you actually get a couple more NADHs. So, a few more NADHs, always good, more electron carriers. Because of all of the electrons being lost in this cycle, we're going to end up with about 6 NADHs from our 2 acetyl-CoAs, but we're also going to end up with 2 FADH2s. Now these guys carry 2 hydrogen ions, so they're able to carry an extra, extra electrons, which is great because we're going to need those extra electrons. They're getting shuttled, by these carrier molecules, these electron carriers, to another stage down the track, which you can see. So the Krebs cycle goes ahead and spins around, and it ends up having a couple byproducts as well. First of all, we get 2 ATP for our 2 acetyl-CoAs. Great. Fantastic. We get an extra 2 ATP. But this is also where our 6 CO2 is created, because what we're going to end up having is a bunch of carbons that need to get used up and a bunch of oxygens that are there to be used up, and the oxygens grab hold of the carbons, and that's the byproduct. So, we flick that off. That's it. We don't need that anymore. So some important information here. We need to kind of remember about acetyl-CoA. As far as I'm aware, this can come up as a question, or at least it's a piece of information that you need to know, that it comes into the Krebs cycle, or pyruvate becomes acetyl-CoA. We also need to remember our electron carriers. Not so much this one here, actually. Not so much this one here. This was a bit of information that I figured out the other day, but definitely that we get our NADHs and FADH2s from the Krebs cycle. Okay, at this stage you'll probably pause the video a couple of times so you can take some notes. That's expected. That's what I want you to do when you're watching these videos. Because we're now up to the electron transport chain. Oh my lord! The electron transport chain, gave so much information when we were coming into it. This is where all those NADHs and FADHs FADH2s are now going to be used. So all of the NADHs come into this cycle, and all of the FADHs come into it as well. And this arrow here is the NADHs from glycolysis. Sorry, my pen is starting to slow down. Yeah, NADH, and I'll just go from glycolysis. There we go. This happens in the cristae. So this is happening in these folds here. Between the two membranes, there's this chain where there's this passing of electrons and this production of ATP. This is the main game. This is where we're going to get all of those beautiful ATP molecules. This stage has another name. It's called oxidative But in terms of what I really want you guys all to remember, I really want you guys to remember that this is the electron transport chain, where electrons are going to be passed through this sequence of proteins in the membrane to go from being attached to NADH molecules to unloading these guys. These guys are going to get unloaded. So we're going to end up with an Na with a bunch of NAD plus molecules, a bunch of FAD plus molecules. But we're going to end up also with a whopping 34 ATP molecules. So this is where the bulk of the energy is going to be made and the bulk of energy is going to be created. This is also where the water byproduct comes into it, because those hydrogen molecules need to be picked up by something, so the water is also being created at this stage. So, important information, again, NADH, FADH comes into this stage, produces the 34 ATP, but we get the water as the byproduct, and we also get NAD plus and FAD plus, these unloaded carriers, and that's what these guys are. They're more of those unloaded carrier molecules. Okay. Oh dear. There we go. Unloaded carriers. So there's the whole process in action again, and I'll just point out up here that when you have the two NADHs that leave glycolysis, they can in fact use energy up getting into the mitochondria through these electron channels. Sometimes some mitochondria let the NADHs in pretty easily, but others really make them work for it. So yeah, they need to in fact use energy to get in, and that's why we get this disparity. Using energy to get into the mitochondria makes it go from, we don't get 38 all up, the 2 from here, and 34 from here. It says about 32 or 34, but yeah. So we end up with a disparity between the amounts that we get. Lucky last bit. We're going to talk a little bit about anaerobic respiration. This is what happens when we have our glycolysis stage happen in the cytosol. So stage 1 still occurs, but we don't have any oxygen present in the cell. So this stage doesn't exist. It's not going to happen. Instead, what we're going to end up having is stage 1 happen over here. So glycolysis happens over here, and pyruvate exists, but it doesn't go from anywhere else. So it stays inside the cytosol, and it goes through one of two processes. Anaerobic respiration is also known as fermentation, because we're going to ferment the glucose inside of the cytosol of the cell to produce either ethanol or lactate or lactic acid. In short, it's plants and bacteria that go through the ethanol or alcohol fermentation. Okay, so these guys go through alcohol fermentation. It means that they're going to instead break down the glucose molecules into ethanol, and then that ethanol can be recycled later on to, when there's oxygen present in the cell, to try and make the energy process happen at that higher yield. In, however, animals inside of our muscle cells, we go ahead and do the lactic acid fermentation, and we create, well, as the name suggests, lactic acid. So it's that heavy kind of lactic acid that makes you feel sore in your muscles, and it doesn't feel that great. If you've done any form of exercise before, you'll know, or you'll remember and will have experienced lactic acid burn before, and it's that kind of heavy feeling, muscle soreness after a hard long run or game of footy, or swim in the pool, or maybe you just had to run really quickly to the toilet. Who knows? You're going to feel some burn. So, like I said, this was meant to be a quick summary of both of the processes. You should start off with this lesson, to be completely honest. For future, I'm going to try and renumber my lessons so that this one comes first. But we went over all of photosynthesis in a very brief understanding. We went through cellular respiration pretty quickly in a brief understanding, and then we also went through anaerobic respiration there at the end in a brief understanding. For any of the two concepts in photosynthesis or cellular respiration, the aerobic side of things, not the anaerobic. Anaerobic respiration really doesn't come up much in terms of all of the processes involved and all of the enzymes involved. Just basically what they are. I haven't even found any questions that ask you for the chemical formula of these two processes, hence why I didn't talk about it in this lesson. I hope you found this one a lot more easy to follow. I hope you found this one a lot more informative. I will try and create something like this, like a brief overview lesson before going into deeper concepts, if those ever occur in the future. And good luck. Have fun.